Right now I'm doing a PCR for my gene therapy project, and I figured what a better time to post an update on what I've been doing in the last couple of weeks or months than right now. If you don't know what I am talking about, you can definitely check out my other videos. I am trying to post regular updates on what I've been doing and on my progress. Bottom line for this phase of my project, I am trying to optimize my gene expression by playing around a bit with the regulatory regions of a gene. By regulatory regions, I mean, of course, the promoter, the terminator region, and other sequences that I am using in my vector. My vector, which is basically a plasmid, of course, will not remain in its current form. This will be like version 1. So in the last month or so, I think, what I did is I transfected my vector using a GFP gene into a specific type of liver cells, which are called HepG2. Basically, what I was trying to accomplish is find out which of the promoter terminator combination gives the best and of course the longest possible expression in these kind of cells. In order to accomplish that, of course, I transfected several of my plasmids, each with its own promoter and terminator, with a GFP gene. This is a green fluorescent protein gene, into HepG2 cells. Then I measured the fluorescence using a flow cytometer. Basically, I wanted to see three things. Number one, the transfection efficiency of my vector variants, as it were. Basically, this means what percentage of cells were transfected using my vector. What percentage of cells are green and what percentage are not green for each variant. Number two, I wanted to measure the median fluorescence intensity of each vector, of course, of each cell given by a specific vector. And I did measure that, the median fluorescence intensity. This will tell me basically the amount of gene expression that I got in those cells. And number three, I wanted to see if my gene expression lasts in my cells. So I was trying to basically measure those cells at specific time points after I transfected them. These results are I'm not gonna try to publish because basically I cannot do this experiment in duplicate or triplicate anymore. However, results are results, and these will definitely help me in the long run and for the projects that will follow with this gene therapy. So without further ado, and if you want more details, let me explain to you exactly what I've been doing and show you what results I got until now. So I don't know how to explain this, but I'll try to explain this in the simplest way possible. So as I told you, I transfected HepG2 cells, which are a type of liver cells, with 24 different plasmids in order to measure, one, their expression, to see which provides the best expression overall, using a green fluorescent protein gene. Number two, to measure their transfection efficiency. I mean, by this I mean, how many green cells will I have out of the entire population? And I wanted to measure the exact amount of decrease. How many times will my expression decrease over time, right? So these are the three sets of data I wanted to acquire, just so I know in the future what exactly is the best promoter and terminator combination to use, right? Which one provides the best expression? So I'm trying to optimize my vector for the best possible expression. I measured the cells. After I transfected them, I measured the cells using flow cytometry, as I said, at 48 hours post-transfection, and I got these results right here. So these are all of my plasmids tested. This, of course, requires some explanation, so let me walk you through this graph right here. These in yellow are the plasmids using the EF1A promoter. But you can see every one of them has a different terminator region. So this has SV40, BGH, and BGA. The next ones, I'm not going to go into detail right here on what I wanted to accomplish, but I tested like 10 different CMV promoter plasmids. But as you can see, these are this is the median fluorescence intensity expression as measured by flow cytometry. To note is that this plasmid right here, so the CMV promoter and the BGH terminator, is basically the same as this plasmid right here. So it has the same regulatory regions, except two things. One, it's linear. This one right here is linear, basically just a linear piece of DNA, while this one is a plasmid, which is circular DNA. And another difference is that this one right here has no antibiotic resistance gene and no origin of replication. Okay? As compared to this one, which, as I said, is a regular plasmid. Here you have the plasmids in orange with the PGK promoter, with the SV40 promoter, and with the minimum PGK promoter. Here you have the control, because of course I measured just the cells with the transfection reagent that I was using, but without any plasmids. So this will act as a benchmark. I know that this fluorescence should be 0 because, cells and transfection reagents have an innate fluorescence, which, of course, I should not measure. And the last plasmid tested is called PCDNA GFP. PCDNA 3.1 GFP is a type of plasmid. It's a plasmid that almost everybody uses in mammalian expression. So this is used for vaccines. I know David and Josiah and Daria use the same plasmid for their COVID vaccine or something like that. But as you can see, this expression right here, it's much more intense than my best expression plasmid right here. So why is this 75,000 and this is like over 100,000? Well, the answer is simple. In all plasmids that I tested and that I built, I'm using cycle GFP. Cycle GFP is a different protein from the one they use in PCDNA 3.1. So this is enhanced. So this is eGFP, and this is cGFP. Why did I do that? Well, because actually I did not have the resources nor time to clone my gene into PCDNA. However, as you can see, you cannot compare their median fluorescence directly because, of course, it's a different gene, so with different fluorescences, but I can compare the decrease in fluorescence. So I know it's not ideal, but what can you do? This is what I've done. So these are the expression for my plasmids at the 48 hours mark. Of course, what I did next is that I measured these plasmids at 120 hours. I did some dilutions and whatnot, but I measured all of the plasmids again, so to speak, at 120 hours post-transfection. So these are the results I got. As you can see here, my best expressing plasmid at 120 hours post-transfection I repeat, in HepG2 cells and in using SMAR vectors, so the vector that I'm using, was provided by EF1A BGH. As you can see, these plasmids right here changed in expression, which of course it's to be expected. And you can see another thing right here, this CMV-BGH linear, which remember it's exactly the same as this one right here, except that it's linear and it lacks the antibiotic resistance gene and so on, has a much poorer expression than the CMV-BGH plasmid. Why is this the case? Well, I think because the cell, because this DNA was linear, it silences my protein, basically, the expression of my protein. So what this tells me is that I should not use linear DNA in the future if I want a good expression, at least. Another thing that I can observe using this graph right here is look at how much the pcDNA expression decreased. So even if the pcDNA has enhanced GFP, as compared to my plasmids, which have a much weaker GFP, you can see how much this expression decreased. And I am pretty sure that this has something to do with the fact that I am using a SMAR region, which is a scaffold matrix attachment region, and pcDNA does not have one of those regions. So pcDNA is just a regular plasmid that they use for mammalian expression. Another set of data that I wanted to show you is, of course, the percent decrease. So let me just show you this graph right here. So the largest decrease in all the plasmid tested in fluorescence intensity at 128 hours was the plasmids that I used that contained the CMV-BGH linear. So the linear piece of DNA had the weakest expression, so had the greatest decrease in fluorescence from 48 to 120 hours. Basically the next biggest decrease was in pcDNA 3.1, because again, this is not a SMAR vector, it's just a regular vector. So you can see this decreased like 6 times. You can see the other plasmids right here. Of course, the percent decrease in fluorescence is not just about the Mars vector, but of course the promoters and the terminators used. But what this graph shows me that is extremely interesting, besides the decrease, the percent decrease in these two plasmids right here, is this plasmid right here. So my best expressing plasmid, which remember it's right here, the F1A-BGH, right, also had the smallest decrease in percentage. So this only, the median fluorescence intensity only decreased 2 times, as opposed to pcDNA, for example, that decreased 6 times. So what I'm trying to say, if I transfect with this particular plasmid right here, and let's say I get a median fluorescence intensity of 100,000 at 48 hours, at 120 hours I will have a median fluorescence intensity of 50,000, as opposed to pcDNA or any other of these plasmids, which will be severely lacking in expression. So this would basically be like, I don't know, 2,000 or something like that, to 20,000 or something like that. So this decreased 6 times, this only decreased 2 times. So just to, I know things are a bit complicated, but I needed to choose a promoter and terminator combination that I'm going to use in the future. Because, as I said, my plasmid will not remain in its current form. But at least right now, I know that in HepG2 cells and using these Mars vectors that I'm transfecting, EF1A promoter and BGH terminator provide by far the best expression and the best expression overall. So not just at one time point, but at different time points in the whole gene expression process. So this is what I've been trying to accomplish. I've been trying to optimize the vector and find the terminator and promoter combination that works best using my vector and in these liver cells. So yeah, hopefully I made myself clear. And if you want to follow along, of course, right now I'm going to switch to a different phase of my project. This was the optimizing phase, and right now I will try to get rid of the ampicillin resistance gene and the origin of replication in my vectors entirely, because study after study has shown that bacterial regions tend to get silenced when we're talking about mammalian cells. So if you want a good expression in mammalian cells using non-viral vectors, as I'm using here for a gene therapy, you must, or at least you should remove the bacterial backbone, as it were, from the plasmid. So, okay, if you enjoyed this project so far, I would highly suggest you subscribe, and of course leave feedback, because this will help me a lot in my next videos. So have a nice day, and hopefully this has been useful for at least some of you.